So my second question was, what was cell-free protein synthesis? So to understand this, you have to understand how a normal cell does protein synthesis. So you need to understand transcription and translation. So in a normal cell, you got your membrane, your nucleus, and all of your DNA. So what happens is RNA polymerase comes in, binds to a promoter, and starts to make an RNA strand. As soon as this RNA polymerase hits a stop codon, it releases the RNA into the nucleus and creates a primary transcript. So it has a 5' end and a 3' end and a bunch of exons and introns. To make it into a mature mRNA, the introns need to be spliced out and a poly-A tail needs to be added. As soon as that happens, the mRNA leaves the nuclear membrane through the nuclear pore and enters into the cytoplasm. The cytoplasm has a bunch of stuff in it, but what we're interested in is all the stuff needed for translation. So what we need is a tRNA. This tRNA has 3 nucleotides at the base, and these are an anticodon. It also needs amino acids. And what happens is amino acetyl tRNA synthetase comes in, and it takes a tRNA and the corresponding amino acid and it binds it together. Then this group moves towards the rRNA or the ribosome. The ribosome has 3 different sites on it: the entry site, the peptide site, and the exit site. And as soon as one comes in, it moves the others over and then it kicks the one off. So what happens is the tRNA comes in, it binds to its codon, which are 3 nucleotides, and then it releases its amino acid and it joins this peptide bond. It forms a peptide bond, and then when it stops, makes it all the way to the end, this gets cleaved off, and then this peptide forms into a protein. Ta-da! Okay, so what is CFPS? It's basically the same thing that just happened, but it's outside of a living cell. So what's really cool about this, and there's a bunch of different reasons that they do this, is that you can actually use it as an open reaction, which means you can take samples, concentrations, and reaction and monitor the reaction while it's happening, where in a normal cell, you have to wait and then you have to lyse the cell, killing the cell, to figure out if it actually worked. You can also, because it's not a living cell, toxicity is not an issue. Because of this, you can work with the expanded genetic code. Simply put, the expanded genetic code is when you make an amino acid that is completely different from the original 20 amino acids. This can be made by making new tRNAs and making new tRNA synthase amino acetals, as well as making new codons or 4-base codons. So basically, to do cell-free protein synthesis, you have cells in solution, you lyse them or break them apart, you get rid of anything you don't want, and you keep the ribosomes and everything you need for translation, and then you add in everything you need for transcription and translation, as well as an energy substrate and anything needed to get the process going, such as pyruvate. When this happens, you add it in, and then ta-da, you get a protein. So this beginning step you can use for different things, and these are called extracts, and one of the most common is E. coli. E. coli is the most common because it's the least expensive, and also you can make E. coli really fast, so it creates really high yields. And to lyse it or break it apart, you use a homogenizer. This is a sample of one way you can break it apart. What it basically does is it pressurizes the cell substance very quickly, and then it stops. It releases the pressure super fast, and when that happens, it lyses all the cells. One problem with using E. coli is that it's a prokaryotic cell, so that it has some trouble with the complexity of proteins that it can create. All these others have plus and minuses to them, but we're not going to get into that. So when you make it to this step, when you add in all this stuff, you add in DNA. So you can either add in plasmid DNA or you can add in linear expression template or LET DNA. So plasmid DNA can only be made in the cell, so therefore it's a lot slower. But LAT can be made by PCR, so it'll be a lot faster. One problem is endonuclease is this little enzyme that likes to eat away at the DNA. So because LATs are straight, it can eat away at the ends as well as in the middle, but it can only eat away at the plasmids at like little parts of it. Therefore, plasmids have a much higher yield than the LATs. One way that they've actually helped increase the yields of LATs is by adding in this protein, which stops this little enzyme from eating the DNA. So this is a relatively new